Hi students, welcome to Year 11 Biology and Module number 3, Biological Diversity. This is our final video in this module and we're going to have a little bit of a look at antibiotic resistance in bacteria. What you need to be able to do for this particular area is to explain modern examples that demonstrate evolutionary change. For example, antibiotic resistant strains of bacteria. Bacteria are a very interesting group of organisms because there are both groups of bacteria that are very important for our survival and also groups of bacteria that can make us very sick and can be potentially deadly. The thing about bacteria is they reproduce very quickly. So we're looking at, often for many types of bacteria, something around a 20-minute reproductive cycle, which means that their numbers can increase very, very quickly. And it also means that we can go through a number of generations very quickly as well. So they're prime candidates for the study of evolutionary change. In fact, the most famous experiment in the history, really, of natural selection is the Lenski experiment. And this has been going on for decades now and has involved, I don't even know what the latest count is, but something like 70,000 generations of E. coli bacteria. They were designed to look at mutation rates around citrate, presence of citrate in the environment as a potential food source. And this experiment has tracked not only the fact that bacteria reproduce very quickly, but they also have very high mutation rates, and they are able to pass any of these mutations very effectively onto their offspring and subsequent generations because of the fact that they reproduce asexually. So that means that the information that is carried by the parent is passed through to the daughter cells. So therefore, any antibiotic resistant gene can confer an increased survival to particular antibiotics, and not only can that individual survive, but it can then pass that gene resistance on to future generations. And this can significantly shift the population, certainly the gene pool. And that's something that we're going to be talking about in a lot more detail when we look at heredity and genetic change in the HSC course. Genes that confer some sort of advantage, if they are preferentially surviving in a population, will then be passed on to the offspring. This changes the frequency of particular genes in the gene pool, and this increased frequency is what has created some real problems around antibiotic resistant bacteria. So one thing we haven't looked at too much is the difference between what is called gram-positive and gram-negative bacteria. And this, one of the main differences that we need to worry about at this stage is just the fact that around the outside of the cell membrane, there is a range of structures that have been built to protect the bacterial cell. Gram-positive bacteria have this very strong, thick peptidoglycan wall. Antibiotics will interact with this particular group of chemicals. The gram-negative bacteria also have the presence of peptidoglycans, but sometimes these are inaccessible, and this can create some interesting challenges for different types of antibiotics. Bacteria, for example, that lack a target for the antibiotic, like chlamydia, don't have any peptidoglycan target for the penicillin, and therefore that particular antibiotic is ineffective against that particular bacterium and that particular disease. Antibiotic targets can also be inaccessible, and so you can see where the peptidoglycan region is in the gram-negative bacteria. Some of the locations of these make it very difficult for the penicillin to penetrate the outer membrane, and as a result it can't get in and bind with the peptidoglycans. And so therefore these particular individuals could be resistant to that particular antibiotic. A third thing is that the antibiotic target may also be modified to prevent attachment of the antibiotic. So it might be able to get there, but it may be modified in such a way. One of the things that can happen with mutations, we can have just enough of a change happening in the three-dimensional structure to prevent the chemical linkage from actually occurring. So this can be a real problem if bacteria are exposed to particular types of antibiotics and certain mutations make them resistant to those antibiotics. This is such a problem that the World Health Organization recently looked at some articles. One of the things that's worth looking at is methicillin-resistant Staphylococcus aureus, and this was a particular strain of bacteria that were causing a great deal of trouble in hospitals, sometimes referred to as golden staph. But these methicillin-resistant Staphylococcus aureus were resisting the attempts of most antibiotics to actually get in and to destroy them. So recently, the World Health Organization have identified a couple of important points about antibiotic resistance. They've identified it as one of the biggest threats to global health, and that in itself is a real concern. It can affect all of us of any age in any country, and it occurs naturally. One of the things that can happen, of course, in natural populations, and this is why we talk about the idea of natural selection, in that there are mutations happening all the time. There is variation in populations, and sometimes that variation is actively selected for by some factor in the environment. And certainly, antibiotics are a selection factor for bacteria in general and for those chemical active sites in particular. One of the problems, though, is some of the misuse that we've seen of antibiotics in humans and also in animals is accelerating this process of antibiotic resistance. So antibiotic resistance would always happen, but the problem is the rapid pace at which it's happening. So what we're seeing, or what the World Health Organization is seeing, is a growing number of infections from bacterial diseases such as pneumonia, tuberculosis, gonorrhea, and salmonellosis. And they're becoming harder and harder to treat as the antibiotics used to treat them become less effective. And some of the reasons for this are around our own use of antibiotics. So that could be oversubscription, so being prescribed antibiotics when we don't need them, or what we'll call as partial use. So that is, I take a couple of antibiotics, I'm starting to feel a little bit better, so I don't take the whole course, I stop part way along. This is also something that can encourage those individuals which maybe would have been wiped out if I'd continued the whole course to survive. They've had that initial resistance and haven't been killed off straight away, and so now that resistance is going to be passed on as they reproduce. And of course, one other thing is the difference between what we call narrow and broad spectrum. In the previous slide, we looked at the difference between gram-positive and gram-negative bacteria very, very generally, and you can look at that in a bit more detail in class. But one of the things about our broad spectrum antibiotics is they are often prescribed when we're not entirely sure of what the cause of a particular infection might be, and they act in a very broad way. So they're going to destroy bacteria in general, whether they're good or whether they're bad, and they tend to affect both the gram-positive and the gram-negative to various extents. Narrow spectrum are much more targeted, and they're designed to specifically look at either one or the other, gram-positive or gram-negative, and perhaps even more specific than that. So narrow spectrum are really those targeted antibiotics that are looking at wiping out particular pathogens, whereas the broad spectrum ones are trying to do a more general job. The consequence of antibiotic resistance is longer hospital stays and potential increases in medical costs, and perhaps even increase in the death rate. Antibiotic resistance is a real genuine concern, and it's a very, very good, though sobering example of natural selection being applied to native populations like bacteria. This is the end of Module 3 and biological diversity. Thanks for watching.